Thank you for joining this live webinar. This is actually the second webinar of the webinar series of 2021, which is organized by the EMN of the Metabolomics Society. My name is Evelina Haridimou from the Department of Biological Sciences at the University of Cyprus, and I will be hosting this webinar with Michelle Reid from Crystal Therapeutics from the Netherlands. I would like to inform you that all the material will be recorded and uploaded to the Metabolomics Society website, allowing you to access the live event at your own convenience.
So thank you again for joining us. If you would like to hear this talk at a later time, like Evelina said, the webinar will be uploaded to the Metabolomics Society website with a link distributed via social media. We encourage you to connect with us online via Facebook and Twitter for future content, and feel free to email us with suggestions or input on upcoming webinars and speakers. In April, we will host independent researcher Maria Eugenia Mongehe and a PhD from her lab, Nicolas Savaley-Gehi, from Symbion Conecet in Argentina. Please keep an eye out on our social media accounts and your email for the upcoming webinar date and time.
Right, so our speakers for today are Professor Assaf Vardi and Dr. Guy Schleyer. Professor Vardi is a principal investigator at the Department of Plant and Environmental Sciences at Weizmann Institute in Israel. The Vardi laboratory aims at exploring the cellular mechanisms and metabolic bases that mediate the interactions between bloom-forming algae and the pathogens such as viruses and bacteria. They study key host pathogen systems that regulate the fate of these blooms in order to discover novel signaling and metabolic pathways employed during these interactions. Newly identified genes and metabolites induced during specific host pathogen interactions are used as functional biomarkers to assess the ecological impact of microbial interactions on the structure of marine food webs and potentially global nutrient cycles. Our second speaker is Dr. Guy Schleyer, who is a postdoctoral researcher at the Vardi laboratory. Specifically, he studies the chemical ecology of marine microbes, aiming to unravel the chemical language that mediates microbial interactions. In particular, Guy combines metabolomics and lipidomics-based approaches in the lab and the field to study the metabolic landscape of host-virus interactions. I think we're ready to start, so I'll pass you on to Professor Vardi.
We just need to share the, we don't have yet the bit. Oh.
Yes, you can share it now.
Hello, everyone. Thank you very much for the kind invitation and for the opportunity to present our work from recent years. I'm excited to be here together with Guy, and we'll give you two talks where I'll highlight first some of the interesting concepts in the lab regarding host pathogen interaction and how can we follow them, not just in the lab, but all the way across scales to the big ocean and its possible implication to the ecology, to the marine ecology. So linking scales between the micro scale when the metabolite is being produced all the way to the large scale of hundreds and thousands of square kilometers in the ocean, that will be kind of the leitmotif of the talk. So let's dive into the cold water of the ocean. And the main phenomena we study for years is this outstanding phenomena that is called algal blooms in the ocean. Talking about single cell eukaryotic cell, photosynthetic eukaryotic cell, that in a a very specialized hotspot can make massive blooms across the ocean that cover thousands of square kilometers. Only recently we realized that half of the photosynthetic activity on Earth is being done by this outstanding ecological phenomena of algal blooms. And you see here nicely the magnitude of carbon fixation in this big process. This is, of course, the foundation of the entire marine food web, and it's composed of this beautiful, intricate, single-cell microalgae that we study in the lab. One of the key questions we study is what is the molecular and metabolic basis for host-pathogen interaction that regulate the fate of this massive bloom in the ocean. And if you wish, this is one of the key ecosystem pathway in which the regulation of this massive fixed organic carbon and the release of it during lysis by viruses and other pathogens is really key to understanding how this flux of metabolites fuel the entire ocean microbiome. So in just one slide to summarize the way we tackle and the way we study it all the way from the ocean to the lab and to molecular mechanism. So basically we're going out to the field, so it's a chemical ecology approach. We're going out on cruises to chase after these blooms in the ocean. You can see these blooms, they are so massive that we can detect them from satellite, from space. And then we are back in the lab with isolates of algae and different pathogens that regulate the fate of these blooms in the natural environment. This algae, as you see here, can be subjected to different pathogenic interaction, mainly with viruses, pathogenic bacteria, chemical communication and chemical defense with predators as micrograzers, but also we study the chemical communication within the same patch, within the same population, and how it's being mediated by what we call infochemical, these chemical cues that are released and can coordinate a population level response. What we really wish to aim is to gain a better understanding on the cellular mechanism that mediate each of these interaction, and what will be the metabolic and the ecological consequence when these cells are being infected by different pathogens. So also to understand basic metabolic principle that mediate this host-virus interaction or host-pathogen interaction, and then once we define and find and map the metabolic map of each of these interactions, we can go back to the environment and using specific metabolic biomarkers that can enable us now to look at this interaction back in the field in the real time in the interaction. So we have this tension between the complexity of the environment and raising robust model system in the lab that we can follow in great detail. So I'll focus from now on one of the hottest topic in marine microbiology in recent years, which is marine viruses. So if you, like you see here in this slide, if you take a drop of water from the ocean and you simply stain it with DNA fluorescent dye, you'll see the big guys are bacteria, but the tiny ones are viruses. Only in recent years, we realized that viruses are the most abundant biological entity in aquatic environment. It can reach up to 10 to the power of 7 viral-like particles in a ml of seawater. They have a huge, not just ecological important, but also biogeochemical, because in their lytic infection, they can recycle and contribute with the release of metabolites to major biogeochemical cycles of carbon, sulfur, nitrogen, and so on. One of the very interesting concepts that was raised more than 20 years ago is the viral shunt. So as I mentioned, huge amount of carbon is being fixed in photosynthesis in these algal blooms, in these massive blooms in the ocean. But when they are infected by viruses, this amount of organic carbon that is being fixed here is not anymore, can be transferred to higher trophic level and instead can be diverted in what is termed the viral shunt and really fuel the entire microbiome composition. So this is a very broad ecological process, and we want now to be able to zoom in into this process and really to try and demystify it, because it's so difficult to be able to quantify. So what we, as you will see now, a few examples, we develop a single-cell RNA-seq approach to be able to quantify active viral infection, because when we look at viruses simply by counting particles, we don't know how many of them are infectious. We don't know what is the level of infection, because it's so complex to watch this and track host-virus interaction in time and space. We also quantifying the genes that are expressed both from the host and the virus in order to identify interesting mechanism of the virus hijacking of the host and resistance in response to the virus. And also we quantify the different metabolites that are being released during this infection. And we combine the single-cell approach with profiling the exometabolome in order to quantify the metabolic landscape that is being produced by viral infection and unique to viral infection. Lastly, as I mentioned, we bring back from the environment isolates of algae, virus, bacteria, and in the lab, in almost a synthetic manner, we try to reconstruct the bloom succession and phenotyping algal bacteria virus interaction in a controlled manner in the lab. Some of the major challenges for you to know from the marine environment, talking about marine virology, aquatic virology, we have a wealth of genomic information from the marine environment, a lot of data set for metagenomics, a lot of interesting discovery, but we're really lacking mechanistic understanding of host virus interaction. And I'll show you some example of that. And also we're missing a lot of information about resistant mechanism and the unique metabolic landscape that is being produced, as I mentioned, during viral infection. And lastly, major challenge is to be able to quantify viral infection, and I'll show you how the single-cell approach can really push it forward, our ability and sensitivity to quantify infection, active infection in the field. One of the amazing discovery in the last decade, almost 2 decades now, from the marine environment, from the aquatic environment, is the presence of giant viruses. You see here in this tree of protists, they can infect algae, they can infect amoeba. Some of these giant viruses can reach, they can contain up to more than 2,500 genes, and also in size, the particle can reach above a micron, micron and a half. So really everything we knew till now in virology that was heavily based in textbook on phages, you know, we have to rewrite again the textbook of virology with these giant viruses that really revolutionized everything we knew about host virus interaction. These giant viruses, you see here also the size of them, a few examples, and can contain metabolic pathway that never seen before in viruses. So the good old days with the HIV virus or COVID, even with 3 to 5 to maybe 7 genes, are very different in the marine environment with these giant double-strand DNA viruses. We specifically study this model system, Emiliania-Huxley, that's the name of the algae we study. It's making a massive blooms in the North Atlantic, and it's not only hugely important, you see this single cell, they are not only hugely important in photosynthesis, they also cover these cells by calcium carbonate shell, beautiful cell with this intricate calcium carbonate shell, so they are hugely important in calcification in the ocean. You see here a relic of these massive blooms over geological time scale with the famous White Cliff of Dover. They're all made by this, by the exoskeleton of this single cell, Emiliania-Huxley. It's also important, and run this video, but these massive blooms are also a major source for DMS, dimethyl sulfide. This volatile is raising up to the atmosphere and can change cloud condensation nuclei. This is the smell of the sea, as you know, but we study the function of this molecule in host pathogen and host-grazer interaction. These massive blooms of Emilia Huxley in the ocean are often infected by a giant virus that we study, Emilia Huxley virus. It's a double-strand virus which contains up to 700 genes. Among them, a very unique biosynthetic pathway for sphingolipids. Guy will tell us a lot about this pathway. This is the only virus to date to contain almost a complete biosynthetic pathway of sphingolipids. Many viruses, as hepatitis C, HIV, can manipulate the host sphingolipid metabolism, but this is the only virus we know to date in biology to contain this pathway. So we study a lot the arm race between the host that also contain, like every other eukaryotic cell, and the pathway that is encoded by this virus. Major concept in the lab is to, as I mentioned, we want to shift between just counting and understanding diversity of viruses to functional understanding, mechanistical understanding. So this is the concept that we like a lot to study and to focus upon, which is the virus cell metabolism. So we're looking at the infected cell or the virus cell as a new entity in which the metabolism is heavily remodeled during infection. And in order to be able to track these changes, in in the lab and the environment and what mediate this cascade of events. So we're looking at active viral infection of the virus cell. Very interestingly, as I mentioned, the virus cell is also, the infected cell is also composed of a lot of interesting metabolic genes that are encoded by the virus and are highly expressed during infection, which can really, you know, we're not just looking at viruses as mortality agent, but also as opportunity to expand the metabolic capabilities of the infected cells. And we study how these new auxiliary metabolic genes encoded by the virus really modify and how they're different from the host. So we study the rewired metabolism and transcriptome of the virus cell in order to decipher different hosts and viral cellular strategies. This is just one quick example of some of the findings we found in our model system of Emilia Huxley and its specific virus, doing a lot of transcriptome, dual transcriptome, and metabolome. We found that reactive oxygen species and a whole network of antioxidants are really important in the activation and onset of the lytic phase of this virus that kill the host in a programmed cell death manner. We also found that autophagy is highly induced during viral infection. You see here this typical double membrane autophagosome that is induced, but in different from mammalian or plant system where autophagy is a defense mechanism against the virus, we found that the virus is hijacking this mechanism in order to recycle membranes, and because lipids are really essential to understanding this host-virus interaction. And also that really minor subpopulation can really resist infection, where majority of the population will crash and induce host cell death mechanism. A tiny subpopulation will, less than a percentage, will induce resistance and change in fluidity. And as I mentioned, sphingolipid and the arm race around it is really major in our understanding, and Guy will talk much more about it. I put here just one example for you to get a feel of the major metabolic changes. As you can see here, we had to develop a very thorough lipidomics platform to understand lipid changes during host-virus interaction. And as you can appreciate, every line here is a lipid, and you see that during a lipid infection, a massive induction in variety of lipid classes during viral infection. Among them is, for example, tags, diethylglycerates, and of course, the glycosphingolipids. Lipids are also important, as I mentioned, TAG. When we look at infected cells, you can see lipid droplets accumulating within the cell. And also, we understood that lipids are so essential because the virion, this is electron microscopy of the virion. The virion itself is composed of at least 2 membranes. So rewired lipid metabolism is really in the heart of this viral infection. When talking about lipids and lipid accumulation during viral infection, and we are a lot of interest in the lab is about understanding cell-cell communication. And what Daniela in the lab revealed a couple of years ago is that these cells are not only produced, you see here the nice virion produced by the cell, but also they can produce high level, almost equivalent to the virus, of extracellular vesicles or exosome-like vesicle that contain in their cargo a unique lipid composition. So that's really allowing now of lipid molecules to travel between infected cell and bystander cell and really modify the cell fate during infection. So we study this a lot, and the cargo of this vesicle Daniela found is not only by interesting lipid composition, but also by small RNA with the potential to remodel the transcription of the bystander cell. She also found, to our surprise, that these released vesicles are actually a pro-viral strategy. And when she added them and then the infection, she can expedite viral infection using this extracellular vesicle. And we proposed it as the new mode of cell-cell communication. Also, when she mixed the vesicles together with the virion, we see that she can extend and alleviate the half-time of the viruses, which are typically in seawater. The half-time is about a day. It's really critical if you have a virus that's really specialized on a bloom forming host, because UV, for example, will affect dramatically the infectivity. But so we study now how this vesicle can really extend the infectivity up to 6-fold when this vesicle meets released virion. Another major shift in the work in the lab is the shift between understanding on the population level, on the bulk analysis level, where when we average the different entities that are different phenotypes that are being produced during viral infection into a single cell understanding in order to really understand the different entities that are being produced during infection. During infection of the virus cell, we can be in early state of infection, we can be in late state of the virus cell. Some of the cell will induce host cell death. Others, as I mentioned, can resist. And also a new finding in the lab, we can also see a coexistence of kind of a chronic infection. So we really want to now to be able to untangle the virus cell sphere to its component, and also how metabolites that are being produced and vesicles as a communication signal can really help us to understand the transition between these states. So shifting from a population to a single cell level, and what is the metabolic landscape that is being produced. So in the last 3-4 years in the lab, and that's very unique to our lab, we develop a lot of interesting approaches to be able to track host-virus interaction on a single cell resolution, starting with automatic microfluidic. Here we can capture a single cell and really wash it with reagent and do qPCR on specific genes of interest. Then we expanded it to more untargeted approach, where now we can do really a single cell dual RNA seq, so both the host transcriptome and the viral transcriptome of each individual cell. And also we combined this approach with a new approach in the lab. This is a new publication from Flora in the lab, postdoc in the lab, to be able to visualize by doing a single molecule fish of a viral gene and a host gene. as a complementary approach to the untargeted transcriptomic approach. Again, this shift from bulk analysis, where we average the entire phenotype, into individuality in host virus interaction. You can see here one example I put. Each of these individual cells, we see here the level of the viral transcriptome. So we see this large heterogeneity in viral expression. And that gives us now a tool to be able to ask the question, what makes this cell highly infected compared to this cell that is completely resistant of the same population, the same isogenic population. So phenotypic heterogeneity. But if we do some just averaging, we'll have just this 1 point here, averaging, and we miss the entire interesting heterogeneity. That also gives us now a very interesting tool bioinformatically, where we reorder the cells, each dot its individual cell and the level of expression, but now we reorder them based on the transcriptional program of each of the viral transcriptional program of each of these cells. So we're not anymore looking just on infection versus non-infected cell, but we can really start to build the infection continuum and really start to define the infection state of each of the individual cells in a pseudotemporal way. So we don't care of which time of the infection, but more about the infection state of each of the individual cell, and we can start to divide them into what we call meta cell, where they shared the same program. This also gave us, now we order the cell. Each column here is a single cell. Each line here, it's the expression profile, viral expression profile, and we order them again now based on their expression profile, this infection state. What you really can really nicely see is that we start to see the cascade of expression program of the virus, and that gave us also a great opportunity to discover the kinetic class of the infection, starting from the immediate early, a group of genes that share the same expression profile early, and up to the late one that are critical for the assembly of the capsid protein. And now for the transition, how can we utilize this approach in the very complex marine environment? So what we did 2 years ago, actually exactly June 2018, in the fjords of Bergen in Norway, we can not only go on cruises, adventurous cruises to the open ocean, but we can have a controlled induction of a bloom in these big bags. We use inoculum from the fjord, so it's a natural inoculum, and we simply add the nutrient in the right timing. We can get massive bloom, and then sample it in a high temporal resolution in order to understand the host pathogen interaction. So exactly what we did. It was a big consortium of many labs working together around the clock and sampled for a different each lab for its specific interest. This is how the blooms look like, really massive blooms. We're talking about 100,000 cells per ml. And what was really interesting is that each of the bag, this is the level of viral production, each of the bag showed different level of viral productivity or viral release. This gradient in viral release, we'll see in a second, was really instrumental to understand some of the unique metabolic landscape. At first, we applied, and this is the first demonstration of single cell approach and tracking viral infection in the ocean, in this case, in this experiment, where the work of Gur in the lab, a postdoc in the lab. This is again the bloom, the crash, and the release of the virus. If you look at extracellular view of the infection, you will see this typical host virus interaction. But what we compile over here is close to 40,000 individual cell for RNA-seq over the different stages of the bloom. And you can get the appreciation of the level of viral expression And what we can really start to watch here for the first time, the pandemic of viral taking over the bloom on a single cell resolution. And for us, as you can see here, and the timing here is we're moving here in during the bloom succession. What is few things that are really amazing here, first, that we can really detect the first individual cells very early on in the bloom that got infected. So Gur and I like to call it the first sip of the Pangolian soup, if we're thinking about COVID these days. But also we can learn about the infection process, but also interestingly about the bystander cells that didn't get infected, in order to understand novel mechanism of resistance, and that's one of the next challenges in the lab. We then couple it with the work of Flora, looking now on specific viral genes and host gene using the single molecule fish, so we can really visualize a transcript within each of the individual cell, as you can see here. And Flora brought us to a very interesting observation that got us very surprised. Even in the peak of the bloom, in the onset of the demise, that we always thought that it's completely controlled by the virus, we got up to 30% infected cells. But still, the entire bloom is crushing in a synchronized manner. So that's raising interesting possibilities. For example, that there's release of interesting metabolites from the infected cells that causing the cell death of the bystander cell, or that other pathogens like pathogenic bacteria work in concert with the virus. And in order to get a better understanding of those possibilities, we couple the single cell work with exometabolomics work, and that's work of Connie and Guy and Nir in the lab. Incredible work that soon to be published, I hope. It's still under review. And what they profile, the metabolites being released into the water on a temporal eye resolution over different stages of the bloom. So each point here in this PCA plot is of over 110 samples across the bloom succession based on close to 7,000 mass features. And what was amazing to see, that really the chemistry and the metabolic landscape can really start to reconstruct the biology of the bloom. So, and where the actual bloom itself of Millania Huxley, we see a kind of homogeneous profiling of the metabolites, but during the demise phase, we see this very large heterogeneity. So how now we zoom in order to see the metabolites that are uniquely produced during infection. And so first, this is another way to look at it over time. Here you see the bloom succession. First, we had another bloom of other algae, and this is the Millania Huxley bloom. And this is clustering of these metabolites. So now, based off this clustering, we can start to look at specific phases of the bloom. As you can see here, at first the first bloom, and then the bloom demise that is unique to Millania Huxley, and the demise phase that is shared between the different blooms, in order really to get unique signature of metabolites. But we wanted to get a direct link to viral infection, and in order to do so, we used the fact that each of these bags or each of these blooms has different level of viral infection. As you can see here, high viral infection and intermediate state to the level of bag 3 that was hardly detected any viral infection. And we correlated to viral markers. In this case, it's the MCP, the major capsid protein. And what we found is unique set of metabolites that is highly abundant during the phase of the demise phase, and was highly correlated to the level of viral infection. Looking in depth, Guy and Connie spent many months in order to understand what is of this molecule, and what we found is very unique metabolic footprint of halogenated metabolites. As you can see here, 17 molecules, and with very unique pattern of chloroiodo molecules. That halogenated molecules are, of course, major interest in natural product chemistry, but this unique combination of chloroiodo is very rare and was very unique to the viral infection hallmark. In these days, we're putting a lot of energy really to understand the biosynthesis of this molecule, looking at different halogenases, both encoded by the host or by the virus, and also trying to isolate this molecule in order to understand its specific activity. Very interestingly, when looking at samples from other cruises that we did in the past, like here crossing from the Azores Island up to Iceland, massive blooms that we found, several blooms on the way, we found that in these samples, we also can see this unique chloroiodo metabolite being produced in the late infection stage of the bloom. So really, we have here a very unique biomarker for infection that now we are very interesting for its function in the marine environment and on the cellular level. Back to the bloom demise, and as we saw in different ways, a very unique metabolic landscape, we are now very interesting to understand how the metabolic landscape that is now very unique to viral infection, how does it shape the microbiome composition? Because very unique metabolites are being produced here, and how and whether the microbiome that associate with the Emilia-Huxley bloom is really specialized on the specific metabolites that are being released here. And in order to do so, we also couple it to 16S and 18S sequencing. And one of the interesting species that we found there of bacteria is Sulfitobacter, is a rosobacter that is highly associated with these blooms and highly abundant in the marine environment. And it's really linked to work of Noah Barak Gavish in the lab, finished PhD a couple of months ago, now a postdoc in the lab, where she found that Sulfitobacter, this rosobacter, isolate from the big cruise I showed you before, can have a pathogenic effect on the same host. She found, as you can see here, this bacteria can really cause demise, but in different time scale than the virus. As you can see here, it attached to the algal cell and can really affect the pathogenicity of the cell when the culture is getting into stationary phase. What was very interesting is that Noah found an interesting trade-off. Strains in the lab that are resistant to viral infection were highly susceptible to the bacterial infection. So we're now examining this interesting trade-off. And eventually, thinking about what we saw in the bloom, that only 30% got infected, but the entire bloom was crashing, we're now looking to whether can viruses and bacteria act synergistically in affecting the Emilia-Huxley bloom. Noah here did this interesting experiment when she had different strains of Emilia-Huxley, both the virus and the bacteria, and we see synergistic effect, as you see here, as compared to virus alone and bacteria alone. Synergistic effect when the bacteria and the virus act together. So now we're thinking of a new concept, not just the viral shunt, as I started the talk with, where viruses infecting lytic virus, the blooms and causing bloom demise, that we're examining whether the unique metabolic metabolites that are being released by viral infection can induce virulence of the bacteria, and then they can act synergistically in order to utilize and maximize the infection of the algal bloom demise. So we call it the pathogen shunt. So now, hopefully, touch wood, end of this year, we're going on a heroic cruise to the Patagonian shelf where massive blooms of this algae occurred, and very important there for the carbon cycle and carbon experts. And we want to be able to disentangle the different pathogenic driver, virus versus bacteria, and to look at the metabolic landscape and consequence the unique microbiome that associated these stages. So I will end with this concept, which is the individual phycosphere. And we want to think that the unique metabolic principle that mediate host pathogen interaction, whether it's a virus indifferent from a bacteria or a grazer, can really modify the unique way of how the metabolism of the host will change and which metabolites will be released. Now, the main challenge is to be able to link between this microscale host pathogen interaction and the big picture of the large biogeochemical cycle, and how this twig between different dominants of a pathogen can really change this link from the single cell level to the big biogeochemical cycle. A good time to stop, and most importantly, to thank the people in my lab that I was so super fortunate to have, especially Guy that is next to me here to continue our talk, Daniela, Flora, Gur, Connie, and Noah, many others in the lab, but those specific one, to the data I presented, our collaborators, importantly our funding, and thank you so much for your attention.
As Guy pulls up his talk, we just want to remind you all to please submit questions, and in your questions indicate who the questions are for. Thank you.
Okay. Do you see my screen well?
Yes.
Okay, great. Yeah, so thank you very much first for having us here. It's really nice to present it to the metabolomics community. So my name is Guy. I did my PhD in the lab of Assaf and graduated about a year ago. And since then I'm doing a postdoc. So I will share with you the work I've been doing in the past few years, focusing on the glycosphingolipid-based arms race between Mediania huxleyi, our algae, and its virus. So as Assaf already showed, we like to look at the infection using this very simplified scheme. So we have a virus susceptible cell. It is infected by a virus to get a virus cell, this special entity. And we see in the lab that following infection, most of the cells die. However, a small population actually survives and evades death, and they become resistant. And of course, we have virions, extracellular vesicles, and different metabolites and glycosphingolipids that are involved in this whole dynamic process. And one of the main questions that interest us, and especially me, is how are glycosphingolipids involved in these infection dynamics and in the occurrence of resistance in the population. So, as Assaf said, there were already several works done in our lab to tackle these, working on the population level, so doing dual transcriptome and looking at primary metabolism. And this showed massive remodeling of glycolysis, of reactive oxygen species regulation, de novo fatty acid synthesis, and of course, phingolipids. And this was also coupled to LCMS-based lipidomics analysis on the bulk, looking at uninfected cultures at different time points and infected cultures at different time points of infection. And this really illustrated different lipid species that are changing during infection. Some are induced, some are reduced, and we could attribute them to virus-specific metabolites, so those that their biosynthetic pathway originates in the virus, induction of host metabolites, and reduction of host metabolites. And I will focus on glycosphingolipids. So sphingolipids are in many organisms, eukaryotic organisms, important lipids. They are structural lipids found in lipid rafts, and they are used for signaling. In host pathogen interactions, they can signal for post-programmed cell death. This is their structure, so they have a sugar head group. They have a long chain base, which is this part, and a fatty acid that is connected through the long chain base. And in our system, we have our host, our algae biosynthetic pathway, and the viral biosynthetic pathway. The virus brings almost all the enzymes required for this pathway, except for two. And what we see is that the host pathway starts with a serine palmitoyl transferase. This is the first rate-limiting enzyme. It starts with a C16-CoA precursor to form a C18 sphingosphingolipid base or long-chain base, which is then processed all the way to glycosphingolipids. Whereas this serine palmitoyl transferase of the virus actually has a different substrate specificity, and it prefers the C15-CoA to produce this unique C17 sphingosphingolipid base, which then continues to viral GSL. So this is the first difference, major difference between the pathways. The host pathway is downregulated during infection, and the viral one, of course, is upregulated. And we also see that if we inhibit the serine palmitoyl transferase during infection, it will actually hinder the whole infection process, meaning that this is a very important process for the infectivity of the virus. So I want to present you now the main species that we have in our system. So first we have, so we divide them to biological categories. We have an HGSL, so a host-derived GSL. It is found in all Emilia huxleyi strains that we checked for so far. It's characterized by a long chain base with 19 carbons, so 18, and another branch one, and 3 double bonds, 1, 2, 3. We have a sialic acid GSL, or SGSL in short. Is characterized by a sialic acid head group and a D18-2 or D18-1 long chain base. And this was found so far in all susceptible strains of EHUX, so all the cells that are sensitive to the virus, and it's actually a very good biomarker for cellular sensitivity of Emilia huxleyi. And we have the virus-derived or the viral GSL that is found only in infected cells because the biosynthetic pathway is brought by the virus. And it has the 17 carbon long chain base, which is unique, and also it has an additional hydroxyl, so it's a 3-hydroxylated long chain base, which is quite unique. It was found to trigger host program cell death in neighboring cells, and it is also found in virions. So these are the 3 different categories that we have, and we look at them in 2 complementary methods. One is comparative lipidomics using LCMS and mass spectrometry imaging. So as Assaf already mentioned before, we work a lot in bulk, averaging the population. But if you consider a flask that is infected, 24 hours after infection, it won't be homogeneous. So many cells will be infected, maybe in different time points after infection. Some cells will already be dead. Maybe some cells will not yet be infected. And maybe some small subpopulation will be actually resistant. And when we do lipidomics analysis of this, we actually average the whole population. So what we ask ourselves is, how can we resolve now this dynamics, this heterogeneity? And the way we chose to tackle it is by using a classical method in microbiology, which is a plaque assay. So the nice thing about plaque assay is that each plaque originates in one algal cell that was infected by one virus. And then this cell slowly infected its surrounding. This is how a plaque assay looks under fluorescence microscope. So in red, you see the chlorophyll autofluorescence of the algae, and all the black spots are different plaques. And if we try to think what would be the theoretical structure of such a plaque, so in the center of the plaque, we will have the original cell that was infected and its surrounding cells that might already be dead. Surrounding them, we will have infected cells at different time points post-infection. Surrounding them, we have this very interesting layer, which we call bystander cells. So these are cells that have not yet encountered the virus, but already get a flow of metabolites from the infected cells, which might change their metabolic state. And surrounding them, we have the layer of naive cells that haven't seen the virus yet and also don't get any metabolic signal from the infected area. And we combine this plaque assay with mass spectrometry imaging using 2 complementary methods. One is MALDI, matrix-assisted laser desorption mass spectrometry, with a spatial resolution of 100 micrometer, and the other one is a flow probe in situ microextraction with a resolution of 500 micrometer. So for each pixel, we get a spectrum of different masses, and using that, we can now tackle specifically lipids and our glycosphingolipids of interest, and we can do also untargeted analysis in order to find new biomarkers that might be important for infection. So I want to show you some results. This is how plaque looks like under the microscope again. This is autofluorescence of the chlorophyll. You see here 2 adjacent plaques at 5 days post-infection. And when we look at 2 GSLs, we can see an image that we expect, that the VGSL appears only in the plaque area, because we know that it will be only in infected cells. And the SGSL of the susceptible cells is found everywhere and is reduced inside the plaque, most probably because cells die. And we can also continue and look at other lipids that are now in our system, so beta-like lipids and other lipids and triacylglycerols. And just by looking at these lipids, we can already see that infection is not binary. It's not a 0 or 1. We can see here different patterns of induction. Some will be induced more in the center of the plaque. Some will start at an earlier stage of the plaque than others. And some, like these TAG, have a very interesting ring-like pattern, which we don't have an explanation for, only hypothesis. And this, when we actually do bulk lipidomics, we average all of these to just say induced or reduced. And now we can see that actually they have different behaviors. And now, using this pattern of induction of VGSL, we actually did some untargeted analysis, trying to find additional metabolites and lipids that behave similar using unsupervised clustering. And we could indeed find many different unknown lipids in our system. And when we annotated them, we saw that for many of them, they share this uncommon C15 fatty acid. So C15-based lipids are known for many years in bacteria in the aquatic environment, but are actually not considered markers for marine algae. And here we show that actually they can be found in algae, and actually they are quite induced, highly induced during infection. And if we put it in the cellular context, so as I mentioned before, C15 fatty acid is actually an important ingredient in the formation of VGSL, because it's a precursor for the C17 long chain base. So it is used by the virus, but it is also found, as we see now, induced in cytoplasmic membrane lipids and also in lipid droplets. And one of the key questions now is whether the induction of these lipids is part of the viral strategy for infection, or maybe it is part of the host strategy to try and fight infection. Additionally, these lipids are not only found in the cells, but are actually found also in viruses and extracellular vesicles that originate in infected cells. And as Assaf mentioned before, this already raises very many interesting questions as to what is the role of these lipids during infection and in resistance. And when we look at the lipid profile of cells, uninfected and infected, compared to vesicles that originate from them and the virions, and we now zoom into GSLs, we can see that actually GSLs are quite enriched in vesicles, both uninfected and infected, compared to their parallel cells, and also in virions, again emphasizing the possible role of GSLs not only as structural lipids, but also as signaling lipids. So until now, I talked to you about infection dynamics, but as I mentioned before, following infection, we many times get a certain percentage of cells that are resistant to the virus. And we were very interested to see if somehow lipids can be involved in this occurrence of resistance. So in the lab, we have many strains of Emilia hackeae that present different susceptibilities to the virus. And you can see here 2 different strains that are resistant upon addition of virus, and 2 strains that are susceptible to the virus. And together with Karmit Zir, a former postdoc in the lab, we had a big experiment looking at 4 strains, 2 resistant, 2 susceptible, with and without a virus, over a time course of 72 hours, where we sampled for untargeted lipidomics using our LCMS, acute of 1. And within untargeted analysis, we got about 12,000 mass features, and upon clustering using k-means clustering approach, we could see a very nice separation between a resistant cluster and susceptible. So what you see here is a principal component analysis, where the first component separates the resistant strains on the right to the susceptible strains on the left. And the second component even separates more between each pair of strains in the TCA. And next, we looked at the differential metabolites and lipids between each of the clusters. And we were able to focus on 43 putative metabolites that are differential. And then applying heat map and clustering, these metabolites very nicely again separate between a resistant cluster and a susceptible cluster, and additionally separate between each pair of strains. And now we wanted to focus on the metabolites that are actually shared between resistant strains. So we have here two main subcluster, one cluster here above that is quite high in one of the strains, appearing also in the other, and interestingly is also induced during infection, and another cluster that is shared between the two resistant strains and is absent in the susceptible strains. And I will give you one example. So we annotated the different metabolites in these clusters and found additional GSLs that were unknown in our system. So in this cluster, we found very interesting GSL with a 3-hydroxylated 18-0 long chain base. So this one appears in resistant cells, but also in infected cells. And this is particularly interesting because if you remember, the VGSL, the virus-derived GSLs, are also characterized by a 3-hydroxylated long chain base, but with a 17 carbon chain. So now we see that actually we get also an 18-1 in resistant cells and also in infected cells. So we looked for different enzymes that can do this hydroxylation. We found about 7 enzymes that appear in Emidiania huxleyi. And when we looked at differential expression, we could actually identify 2, number 4 and 5, that are differentially expressed in the resistant strains compared to the sensitive strains. And one of them may be even a bit induced during infection. And if we now look at their similarity using a phylogenetic tree, very nice, we see that actually they group very close together to the viral hydroxylase. And this raises many interesting questions as to the coevolution of these metabolic pathways in resistant cells compared to infected cells. So another GSL that we found was shared between the two resistant strains and was absent in the susceptible strain. We therefore term this GSL a resistant GSL or an RS GSL. It has a long chain base of 19 carbons and 4 double bonds. Again, 4 double bonds is quite unique in the marine environment, and only recently it was described in other microalgae. And this is for us now a very good marker for resistant cells. And also here we could find 5 different desaturases, long chain based desaturases in Emidiania huxleyi, and out of them 2 were differentially expressed in resistant strain compared to susceptible or infected strains. And once again, one of these desaturases closely grouped with the viral one, again emphasizing the interesting coevolution of these metabolic pathways. So we have now 4 different categories of GSLs, one that appears in all cells, host one, susceptible one, virus-induced one, resistant one. And of course, we always are interested to see what is the relevance of what we find in the lab to the natural environment. So I go back to the mesocosm experiment that Assaf presented before that was done in the Marine Biological Station of Bergen, where we sampled 4 different bags that were inoculated with natural population. As Assaf said, we had blooms of huxleyi in all the bags, but they weren't the same, they differed. So what you see here is the counts of Emidiania huxleyi. We had bloom and the mice, and the viral proliferation of the virus. And bag 3 was the least infected one, and bag 4 was the most infected one. And of course, what we did, we developed a comprehensive chemical profiling of both intracellular and extracellular metabolites during bloom succession. And this is a joint project with Constanze Kulisch. And here you can see an example setup that we had for all our solid phase extraction work. So this is a general layout of our work. I won't go in details over it, but just in general, we had 1 to 6 liters of water from the bags or the fjord. We filtered them with different filters, and these were then extracted, and we extracted the filtrate, either acidified or not, with solid phase extraction cartridges, Oasis HLB by Waters, for also different analysis. And I will focus here on 2. So intracellular extraction of 1.6 micrometer filters that was then injected using a C8 column for lipidomics analysis, and an extracellular extraction of non-acidified filtrate using SP cartridge, which was also injected in C8 column for lipidomics analysis. So of course, we want to look in our GSLs. Intracellularly, we see nicely HGSL, and this was already reported in a mesocosm from around 10 years ago, very nicely showing the growth and demise of Emidiania huxleyi. And what was quite astonishing for us is that we don't see HGSL only intracellularly, but we also see it extracellularly, again, quite fitting the demise of the bloom and the different dynamics. And the same for VGS cells. We could identify around 7 different VGS cell species, and they very nicely repeated the viral counts, the viral proliferation that we had, with bag 4, the most infected one, so the one that showed the most VGS cells, and bag 3, the least. This is intracellular. And when we looked extracellularly, we were quite interested to find that actually the levels of VGS cell extracellularly are in the same order of magnitude as the intracellular levels of VGS cell, again, showing the possible role of GS cells in signaling, not just intracellularly. And of course, we were interested to know if we see any resistant GS cell, and unfortunately, we couldn't detect any in the mesocosm. However, we did collect different isolates from the mesocosm. Some were resistant to the virus, some were susceptible. And when we profiled the lipidome of these strains, we could see that susceptibles matched susceptible strains from the lab, and the resistant isolates from the mesocosm indeed have RSGS cells and these T18-0 GS cells. So they had an identical lipid profile to the resistant strains that we had in the lab, which means that they were in the water during the experiment, only they were in such low numbers that they were below level of detection. And lastly, something very interesting that we observed in the lab, so we have susceptible isolates that we collected during the mesocosm, and for many of them, when we infect them, they die, but shortly after they recover, and we get a population that is now resistant, as we saw in lab strains. And when we now compare the lipid profile of the recovered population to the profile of the resistant one, we actually see that they are quite identical, meaning that the recovered cells now also gain these RSGS cells and the T18-0 GS cells, although they started as susceptible strains. And this raises now many interesting questions regarding, is the population in the beginning has a small subpopulation that is already resistant, and by killing the susceptible population, we allow the resistant population to survive and grow? Or is it indeed an induction of resistance in cells that are infected? So the infection itself allows a shift of the cells from being susceptible to resistant. And this is now one of the centers of the research in our lab. And of course, we're still asking what is the functional role of these RSGS cells? If we now isolate RSGS cells and add them to susceptible cells, will they become now resistant? So I want to summarize it using a very general scheme for our system. So looking at glycosphingolipids, we focus on the long chain base, and we see that long chain base really differentiate between the different biological categories that we have. So a susceptible cell or a susceptible population will have SGS cells and HGS cells. Upon infection, it will start producing VGS cells, and of course have still the susceptible ones. And then we have a shift in the population, and we get the resistant cells, and these cells will now produce RSGS cell. They will produce GS cells that, similarly to the VGS cell, have a 3-hydroxylated long chain base, and of course they will have the general HGS cell. And all these GS cells are also reflected in the rheons and the extracellular vesicles that these different populations produce, suggesting their signaling role in this dynamic process. And I will end with the same slide that Assaf started. So we like to look in the lab at different processes across scales. And in the GS cell case, we look at GS cells that are extracellularly in the water, inside vesicles and virion, in different strains and isolates of cells that we have. We look at their dynamic induction or reduction using plaque assays. We look for their relevance in the natural environment using mesocosm experiments. And in the past couple of years, we joined the TARA project, and we now have the possibility to go to the open ocean, to real blooms of EHACs, and look at these GS cells and try to understand the population composition by using these very interesting biomarkers. And with that, I will finish and thank Assaf a lot for all the support along the years, Constanze Kulisch and Karmit Ziv, with which the work was done, and other members and partners of the Vardi lab. the Mesocosm team, and our funding agents. And thank you very, very much for listening. That's it.
So thank you, Guy and Assaf, for two very engaging talks. And the floor is now open to questions. And so we'll wait as those come in. But in the meantime, I would like to ask you, Guy, about your Nature microbiome paper. Did you see a difference in the identified lipids using FlowProbe versus MALDI? And did you also try, or are you continuing to try any unique techniques like OzID or introducing ozone into the MALDI source to help with lipid identification?
Yeah, so there are differences. Let's say the lipids that are most common, we see nicely in both. The FlowProbe one, so which was organic solvent extraction, let's call it, had more wide range of metabolites. We could see small metabolites like DMSP, dimethyl sulfonylpropionate, for example, which is an important metabolite in our system. I would say the MALDI captured mainly lipids, whereas the FlowProbe had a bit of a wider range. But let's say patterns and induction reduction were quite similar between them. We did try several approaches to look not only at lipids but metabolites, so using some washes to wash out lipids. We didn't get that good results, and I think a year ago we also tried to do MALDI2, which we have in collaboration with our lab to increase our sensitivity, but this is still ongoing.
Thanks.
I also have a question. You talked about, Vardi talked about lipid droplets. How do these lipid droplets benefit the pathogens? I mean, I know that these lipid droplets are essential for hepatocytes, let's say, but how do these lipid droplets differ from mammalian cells, and how do they benefit the virus from evading, the host?
So many viruses are using lipid droplets as part of their life cycle. In a host, like in mammalian, of course, hepatitis C, dengue, and also in our case, that's a storage for fatty acid for the virus to build its lipid membrane. But also, if you keep intracellularly free fatty acid, it's also cytotoxic, so it's also a sink for free fatty acid to be localized. And in many cases with viral life cycle, this is a major source for the lipid membrane. And as I mentioned, autophagy is a major process during infection. So we think in part it's the mechanism to recycle lipids from the lipid droplets in part. The actual composition of the lipid we wanted, but we never did the isolation of lipid droplet. In general, in algal research, there's a lot of interest in lipid droplet as a source for neutral lipids, as a source for alternative energy. So there's a lot of biofuel work on lipid droplets from algae, but we didn't get into great detail of that. And also.
So the viruses are also so they're also using it as a source as an energy source, at the same time as using other metabolites as energy. But do you know if these lipid droplets are the main energy source for these pathogens, or do you know if they're using other metabolites to survive and replicate?
But keep in mind that the host is photosynthetic, so which will produce glucose and other carbohydrates. So at least at the first stage of infection, they utilize the intermediates from photosynthesis. But then a later stage, what we see is the genes that encode for photosynthesis are completely shut off, at this case, and we see many genes involved in fatty acid oxidation, beta oxidation are induced. So it might be that at a later stage, this is how the virus is being energized to complete its life cycle when the chloroplast is shut off. Yeah, it's possible. Yeah, that's one of our major hypotheses there. Also, the accumulation in lipid droplets of nucleolipids, we also see it in the extracellular vesicles that are being released. They're highly enriched in tags.
All right, so they also release this in their extracellular environment. That's pretty interesting. Wow.
And the virus is also encodes for lipases, so it's another probably mechanism to release free fatty acid from the nucleolipids, from the tags.
Yeah, and to follow that up, I would want to know, to what degree are you able to monitor when you work in the environment, this cross-feeding that happens between lipids that are produced, released into the environment, and then taken up and incorporated into the host through virus? How do you monitor that? I'm imagining it's impossible when you move from bulk to a marine setting, but is there something that's being done to cultivate this?
So that's exactly one of our efforts now, to look for bacterial isolates from the bloom demise that can specialize on the molecules that the guy showed us now so nicely on the viral-derived glycosphingolipid, if there are bacteria that can utilize it as a sole carbon source. And we're doing enrichment experiment with microbiome from the environment, diverse bacteria, and we spike them with the viral-derived glycosphingolipid, and we watch its, you know, if we have specialized enrichment in specific bacteria that we try to isolate now. And so that's one way to see whether it's being utilized.
Yeah, I mean, in the mesocosm itself, we did, I mean, we had the ability to do both intracellular, extracellular, and also Daniela from our lab, she isolated viruses and vesicles from the mesocosm. So on all of these fractions, we could then do metabolomics and lipidomics, and then see, for example, which lipids we see intracellularly, extracellularly, in virions, in vesicles. So not directly the shift between them for specific ones, but let's say the accumulation and the dynamics throughout the whole process.
Okay. And as you monitor the dynamics, do you envision it becoming possible to replicate what is happening in environment through labeling experiments? Or is that
we're thinking of labeling, isotopically labeling the viral-derived glycosphingolipids, and then to see its accumulation, it's released through a viral release, because also the virion are enriched with them, but also into the vesicles and to the bacteria that associate with cells.
For example, nanosims could be a nice avenue to do it. Yeah. To label cells, get viruses, and then see incorporation into other microorganisms.
Yeah.
So on that, how essential are these glycosphingolipids? Like, if you block their synthesis, do you actually kill the pathogen, or can they survive without synthesizing these glycosphingolipids?
So when we inhibit the serine palmitoyl transferase, so the first enzyme of the pathway, the infection process is hindered. We don't get readily infective viruses, so it is crucial for the infection.
And it is kind of
for the infection, yes. But the virus still survives. The virus will survive without infecting the host.
It's a question of what you consider virus survives.
Can it still replicate
itself without this glycosphingolipid? So
right.
When we block the pathway, we don't see viral release.
Right. Thank you.
but also when we block, for example, de novo fatty acid synthase, it's similar. When we block autophagy, it's similar. When we block ROS accumulation. So we have different, we can block the release of the virus by different metabolic pathways, inhibition of different metabolic
pathways. Guy, I have a question from, again, your work that you showed. You have this really nice image of tag 48:1 that is exquisite. And so I'm wondering, with the bilayer that you're seeing, well, yeah, I guess tri-layer, the multiple layers that you're seeing in virulence, do you think that this tag could be an indicator of the processing of virulence, or telling you, I guess, which stage of virulence, just because of how distinctive it is? Or do you suspect that it's a different mechanism that is actually going on within the virus?
So we have different hypotheses. One was related to photosynthesis, because the tags, they have different, so they will be processed differently under light and dark conditions. So one option is every ring will represent, for example, a day. So similar to years in a tree or something, this will be days. This is one option, but of course it has something to do with infection, because we don't see it everywhere. Another thing that we were thinking about is sort of ways of infection. So we think of infection as something that is continuous maybe, but we know also from bug experiments that they are usually cycles of a day. So cell lysis will occur usually during dark. So one option is that essentially we see here the middle, the original infection. One day there was one ring, another day there was another ring. And then in this case, tags behave different than the other lipids, which we see as a continuum. But so it's easy to hypothesize here. It's hard to prove it.
Yeah, but I mean, it leads you to many different, like you said, hypotheses. But I mean, working with something like nanosims, as you suggested, would help in understanding this, and then doing a controlled experiment, like you suggested, of light exposure would also aid that. But
I mean, another thing we do, which we didn't present, is actually before doing the MALDI to sample the plaques for viral presence, so using qPCR. So we can sample different areas of the plaque, do a quick qPCR using the viral major capsid protein, and then at least know, because we show lipids, we assume that VGSL corresponds, or we know that VGSL corresponds to infection or viral presence, but we don't know, let's say, if the cells next to it are already infected with the virus, just not yet producing VGSL, or they are not yet infected. So by doing qPCR, at least we can see if there's viral DNA there, and then a bit better to connect the status of the cell to the metabolic state.
Okay.
Okay.
I think that's it. I think we don't have any more questions. Michelle, can you see any more questions?
No, I don't see any more questions. This is your last chance, audience members and attendees. So I don't mind then wrapping it up. I want to really thank both Professor Vardi and Guy for presenting really intriguing research and for your talks today. And we look forward to having the audience in future webinars. Thank you so much.
Thank you very much.
Thank you both.
Have a nice day. Bye-bye.
Bye. Bye-bye.